Random versus directed mutation. Geneticists have wondered whether mutation is a random event or is directed by environmental cues. Two classic experiments in bacterial genetics support the random nature of mutation. In 1943, Salvador Luria and Max Delbruck compared the frequency with which bacterial cells resistant to bacteriophage T1 occurred in a culture. 20 individual liquid cultures of Escherichia coli were transferred onto petri dishes containing a dense layer of T1 phage particles. Variation in the number of resistant colonies per culture, each presumably arising from a single resistant bacterium, led the researchers to conclude that resistance was the result of random mutation rather than a change directed by the presence of T1 phages in the culture environment. In 1952, Joshua Lederberg and Esther Lederberg demonstrated that mutant bacterial cells can arise prior to growth in a selective environment. Cells were grown on petri dishes in a non-selective medium that contained all necessary nutrients but no antibiotics or phage particles. Cells were then transferred by replica plating to plates that contained a selective medium. Replica plating is performed by touching the surface of a plate containing bacterial cells with a sterile velvet cloth, and then touching the cloth to a second sterile plate. Replica plating transfers colonies from one plate to another while maintaining their relative positions. The Lederbergs found that mutant colonies that grew on the selective medium always occurred at the same position on multiple replica plates, leading them to conclude that the mutations arose before growth in the selective environment. Mutation events, therefore, are random and are not directed by specific environmental conditions. And that is random versus directed mutation. Have a great day.